I think that will make it better and more, yeah. So, yeah. So good morning, everyone. I would like to say a bit what I would like to do today. So this is the very last 2 hours. It's a bit, if you wish, a wrap-up 2 hours, where in fact in the brochure I had proposed to call it classics. So basically that was the title, and I would like to motivate why it was the classics. So the main point is if I'm recapitulating what we did since the beginning of the week, and you all know, but I started by inert passive motion, KT. This was day 1, observation of particle. The second day was introducing some active matter, living matter. We started to have very stimulating discussion yesterday where what was the frontier between active matter and living matter. If you have active matter, is it a living matter? If you have a living matter, is it an active matter? What is the frontier in between? And I was proposing that somehow the word from KT to 10 KT, basically, of ATP hydrolysis, allows to, thanks to molecular motor and directional motion, encode most of the biological activity that are essential for having life. So activity is essential to eventually having living systems. This is at the core of living systems. And then the second day in the afternoon, we went to the lab. And again, thanks to Pramod, thank you, we could have the pond water. And then we have the shared place where in the Google Drive, you can see the motion of all types that we had. They are indeed directional. You could see bacteria going like very, very straight and then changing gears. You could see Volvox that was rotating, all kinds of motion that was really directional. And by definition, if you wish, require energy. So there is energy which is ATP hydrolysis driven. So that was the second day. And so my intention was to say, well, inert matter is a given in the living matter. And so the living matter has a strategy to overcome the inert matter property by having this energy source and this directionality. On Wednesday, what I was introducing is saying something like, let's be fair. What I talked about on Tuesday was essentially individual biopolymer, individual molecular motor. I was not yet at the scale of a cell. And as you remember, in the first day here, I was drawing a scale where the polymer scale was nanometer, whereas the cellular scale was micrometer. So on Wednesday, I was trying to say, you need to go to higher order. And for this, I was quoting the paper from Anderson, with the title, which is More is Different, which is a very nice title because it says more is different. It's if you have an ensemble of many, many protein or many, many particles, in concert, they will not act as the simple addition of the individual particles. If you want to remember this idea very, very quickly, one way to go is to think about a crowd. When you are in a crowd, you don't move the same way as if you would be alone. What I'm saying is obvious. There is nothing original in what I'm saying, but somehow it's a way to remember that collective effects are essential. And in a cell, it's very rare that the molecular motors act alone. Most of the time, they are 10 to the 3, 10 to the 5, 10 to the 6. It's a gigantic assembly of many, many filament and many molecular motors. So immediately you come in the reality of collective effect, and more is different. So that was the third day. That was on Wednesday. And then I was saying that the cytoskeleton elements are one of the most important. Why? I was talking about the microtubules, about actin filament, and I was focusing on microtubules because that was a way to illustrate that you can write the equation of the dynamics of the microtubule, the dynamic instability. You can find a function to this dynamic instability, which is essentially permitting the cell to find its center through the centrosome and the Zenga game that one of you was finding. Basically, it's allowing, if I'm the centrosome, I could find my center if all the wall of this room is a cell wall. And then I was continuing the presentation saying that, in fact, these microtubules are all over the place. And in particular, the swimming microorganisms that you saw, you know, the swimming bacteria, Volvox, in fact, all of them have microtubular structure, not individual. These are flagella that are doing this, and in some of the cases, you also found, I think, a helical pitch that was, you know, a very funny bacteria that was doing this. And again, please share all your movies on Google Drive. I think most of them are already there, but put them all. And so the idea is that somehow of saying, well, fine, they have some of them 9+2 microtubule, which are perfectly organized in the flagella, with dynein motor, which are in between, and that somehow are allowing as a whole to constrict and to allow the flagella motion as a whole. So again, a collective effect, right? It's an assembly of microtubule plus dynein motor, which are allowing the flagella to do this type of motion. So it's a real collective effect, and you see that it starts having a link between what you observed on Tuesday afternoon. Then on Wednesday afternoon, the idea was to say, well, fine, fair enough, we have this motion, but let's try to characterize this motion. And for this, I introduced you to, and many of you knew already, Fiji. So basically, you were uploading your movies of Monday afternoon, your movie of Tuesday afternoon. You had the particle jiggling around, or you had the microorganism moving straight, etc. And I was telling you, if you are a dedicated experimentalist, you have to calibrate your system, point number one, micrometer in scale and temporal in time. And once you have calibrated it, you have to track the center of mass of your particle. So many of you have started to do it. I invite you to finish and to contact me. I'll give you my email coordinates. You can send me email. I'm happy to answer. Basically, if you take a particles, if you measure the trajectory of the particle, if you plot the mean square displacement, you should have a linear behavior with time. And some of you, I think you had it, and another group had it also. And you can check the Abchig relation I was presenting day one. And if you track the particles moving day two, you know, the swimming organism, you're going to have all kinds of behavior. You're going to have you know, motion like this, and then change of direction, then change of direction. And in principle, you're going to have a different mean square displacement. So the punchline of this Wednesday afternoon, hands-on, was the way of quantifying your data of Monday and Tuesday. So far, so good? Okay. Yesterday, and that's the last part of the summary I wanted to make before coming to today. Yesterday, what we saw was the fact of saying, well, let's be honest, this motion of microorganisms are not only active matter. There is adaptation to the environment. There is a signaling network happening. There is a more profound processing of information that needs to be taken into account. And what I was writing here is the systems biology or the synthetic biology, which was a way of saying that all the molecular scheme of interaction, you know, there is a receptor, a ligand binding a receptor at the surface of the membrane, which is going to transduce information to another step, which is going to phosphorylate a protein, another protein, etc., is going to be the way the information of the cell is processed. So in addition to having this active matter property, one has to have in mind the regulatory pathway that cells have, because that's biology, and that's the regulation that the cells are using eventually. And for this, I was going slowly through the arguments. Systems biology is a field in its own right, which is expanding at the international level throughout, and with a key contribution from Indian scientists as well. But in fact, the idea is that now you can have genome sequencing. We will have our genome in our generation, and there is a need for many contributors, in particular from any field, to make a link between the different genes and their regulation. So that's the world of systems biology. At the core of the information processes, there is the dogma of molecular biology. DNA gives RNA, gives protein. And if you have this dogma in mind, then you can write the differential equation associated to this DNA gives RNA, gives protein. These are differential equations I was writing on the middle board here. And as you remember, it was very easy equation to pose. It was like the variation as a function of time of each of the species. There is the species that are coming in minus the species that go out, like the microtubule plus and minus, right? It's a real, like you count what's in and what's out. Of course, it can be much more sophisticated than what I'm saying, but the core of the equations usually are of that type. And then I was posing to say, okay, fair enough. This is for the information processes DNA, RNA, protein. It can be set into equation with constant, kinematic constant that can be written for each of the reaction. But let's be honest, genes interact with each other. So it's not only one gene that is going to express a protein, it's going to be a gene that is going to activate another gene, inhibit another gene. So already with 3 genes, or 3 gene, RNA, protein in interaction, it starts to be a significant problem because then you have 6, if I'm simplifying already, a potential differential equation if you have only RNA protein. And that's what I was writing here on the bottom left in the middle board yesterday. And I was making a link between all this story and the motion of bacteria, quoting this paper, where in fact they were saying, okay, fine, whenever there is a sensor coming, a receptor and ligand coming, the tumbling frequency of the bacteria is going to be affected, so either it's going to tumble like this or it's going to move straight. So the information processes go through this pathway. And the last element which led us to yesterday afternoon was taking a very simple synthetic network that was proposed some time ago by Michael L05 and Stan Liebler, which was saying if A inhibits B, inhibits C, inhibits A, I might have oscillation. And one of you was very cautious. Basically, it might, right? It might, it maybe oscillate, maybe it will not oscillate. And I like this because it's a way of saying that I can hand wave the argument, but basically I will never be sure that the oscillation is going to occur unless the simulations are run. And I hope I convinced you about this because this is an important message that for all this calculation, you cannot avoid the equation, basically. You have to face them. You don't need to solve them. But at least you need to be sensitive about the fact that you cannot bypass this equation. They have to be posed and they have to be solved. And solving the equation requires attention, concentration, time, etc. And in the afternoon, I was sharing a simulation that was prepared by one of my colleagues in Strasbourg, Morgan Madec, who prepared the Google Colab that I think you were connected to, or some of you used the Copasi. And basically, I was asking you to trace the phase diagram that they were having with the alpha and the beta. And you saw that, well, sometimes it oscillates very nicely, ABC, sometimes, well, no. And sometimes it's ambiguous. We don't know if it oscillates or not. So the so-called phase diagram tells you that for a selection of set parameter, you will have oscillation or you will not have oscillation. So it's not an easy problem. And for those of you who run simulation, and there is one prominent simulation person in the room, you can share your experience with the others. It's not like it's a real work. It's like an experimental work. You need to design very carefully your simulation. You need to scan parameter. Sometimes it's working, sometimes it's not working. You have no clue why. You need to start it again. The intuition is very bad for this type of simulation. Sometimes you understand, sometimes it's really like an experiment simulation. So at the end of yesterday afternoon, you were showing, several groups who were showing their results, and I invite you to continue to run it. It takes time, I know, but at least you will get a feeling that running the simulation is an exercise in its own right. And funnily enough, the simulation people call their simulation experiments. If you talk to real simulation people, they will say, oh, let's run an experiment. When you're experimentalist, you say, it's not, you're not at the bench. But I don't know if you call it experiment, but my colleagues call it, let's run an experiment, simulation experiment. So I was showing this part just a way of saying that, okay, we have already 3 genes. It's already simple. It's a synthetic biological network. It's working because I was showing at the very last 5 minutes this oscillation that was reproduced 16 years after the first RPC letter. And you saw the beautiful color of the bacteria. They were turning red, yellow, blue, red, yellow, blue, and it was a perfect oscillation. So they had tuned the parameters so that it was working. So, punchline, regulatory pathway, system biology with a network organization is an efficient way for the cell to regulate its state and to regulate its potential fate. So it's a real element that is embedded inside the physics of active matter. And what I'm trying to share, the idea is that somehow these 2 fields of physics should not be seen as having their own life. It's this, if you wish, the same, it is two sides of the same coin, right? Either you take a look at the active matter or you take a look at the system biology part, but it is essentially two sides of the same phenomenon. So far, so good? Okay. Any questions, comments, or it's okay? So you see the logic of the lecture, right? Since moment. So today, what I would like to do is the following. In fact, there is a trend nowadays in science, basically, to consider that the paper that was published last week in Nature is a revolution, right? I'm exaggerating, obviously, but there is a usual trend to fact of considering that the latest work is necessarily very original. I think this is wrong. Basically, I and many of my colleagues actually consider that it is wrong, and this is what I would like to share in the word classics here. So the way I was building this last session is to share two papers, old, voluntarily old, basically, and to share the idea that this old paper, if one reads them very carefully, most of the ideas that I presented, which have been revisited with very original features, it's not the point, with very clear features, most of these features are already written in this old paper. And what I would like to share with you is my personal enthusiasm to read these papers, basically, to go slowly through these papers, and eventually to reread these papers. Without connection to internet, without movie, without everything, just in a quiet environment. Maybe you are used to the sound of the campus, but I'm amazed by the quality of the music of the birds. I mean, when I wake up in the morning, so you go in the middle of the campus, you have only this text on the paper with you, and you listen to the birds and you read this text. I advise you to do that, right? Disconnected from internet. So you will see what was said. It's per cell. I selected one paper for the first hour, then we'll be having the pause. And then there is another paper. The first paper, I'll dwell on it, the second paper, which will be much quicker. But basically, it's classics in a sense, like classic in literature, where you have, these lines, you can read them 100 years after, you still, can think about them. So that's the purpose of now, okay? And at the end, I was asked to give more information about the master and the link to contact me back. So in the last 5 minutes, I'll give you this, but this would be at the very, very last 5 minutes before the photograph, okay? That's the menu, it's okay? Good. So I'm starting by the paper. So this is per cell. You see the title. Rings a bell? Life at low Reynolds number. So basically, Purcell write this paper. He's in Harvard University. He write this paper in 1976. So a while ago, right? It's like an oldish paper, but you're going to see that it is remarkably clear. There is essentially hand drawing, and it will answer some of the questions that you were asking. You know this paper already, or? You've heard of, so I think I've been taking out some of the excerpts, and then I'll show you also, one speaker who is describing the paper. And I'll ask you a question in the same way I was doing so far. So they have kept the oral style. So the point that they wanted is that it's a tape. So I'm reading here, this is a reprint of a paper of the same title that appeared in this book. The personal tone of the original talk has been preserved in the paper, which was itself a slightly edited transcript of a tape. So, a magnetic tape at that time was the way to record. The figures reproduce transparencies using the talk. So there is no fancy figure. It's essentially hand drawing of the figure. The demonstration involved a tall rectangular transparent vessel of corn syrup. And this I will show you. On YouTube, there is Berg showing the experiment so that you are convinced that this is correct. And then projected off by an overhead projector turned on its side, some essential hand waving could not be reproduced. I mean, at that time, you didn't have movie camera that common, so basically they were doing essentially the text record. So I will pass the introduction. It's more a personal vision of the speaker, actually. And then he introduces what I was telling you the very first day. I'm reading here, and again, I will ask you a question in the way for the moment I'm reading. Now I'm going to talk about a world which, as physicists, we almost never think about. The physicists hear about viscosity in high school when he's repeating Millikan's drop experiment, and he never hears about it again, at least not in what I teach. And Reynolds number, of course, is something for the engineer. Basically, Reynolds number is a way to estimate, as I showed you the first day, the ratio between what and what? And the low Reynolds number regime, most engineers aren't even interested in, except possibly chemical engineering in connection with 3D size beds, a fascinating topic I heard about from a chemical engineering friend at MIT. So it's a really oral text, right? Written like he's talking to a friend around a beer or around a juice, right? Basically, it's a relaxed discussion. But I want to take you into the world of very low Reynolds number, a world which is inhabited by the overwhelming majority of the organisms in this room. So, you know, pond water and etc. This world is quite different from the one that we have developed our intuitions in. I had one person came in yesterday and saying, well, is it still physics? Yes, well, so basically it's exactly what he's saying, that somehow, yes, it's physics. But the point is that it's a different physics. And that's the whole point, is that at low Reynolds number, you need to break everything one has been used to and think differently. And he's aware of it. So that's what he's going to proceed to say. 1976, right? So I might set what got me into this, so then he's going to proceed to say, to introduce something that will come later, I'm going to talk partly about how microorganisms swim. So we are really on the second day of experiment. That will not, however, turn out to be the only important question about them. I got into this through the work of a former colleague of mine at Harvard, Howard Beck. And I found a video of Howard Beck talking. So you will see him talking and expressing which experiments he did. And it is the bacteria experiment swimming in chemotaxis. So you will see the link. I can pass a little bit because this is not essential. And then you're going to see the figure 1. Figure 1 is this one. Do you recognize something here? Yes. So the first element he's starting to do is to estimate the Reynolds number, where he had an object of size A, whichever object that is, and then there is a viscosity of the medium, density of the medium, and it's moving at a given velocity v. And so once he has this, he can have the ratio, which is the Reynolds number, and he has essentially what I was showing to you the other day. It's the size of the object, the velocity at which it's moved, the density over eta. I wish you can give, I will speak under the control of Manoj, but when I have a student who gives me a scheme handwritten, I like, because somehow it's personal, it is a handwritten, it's not through a software which is imposing shape, so at least I see a little bit who is writing, who is talking. And I like that, because then, you know, it's personal, right? So basically here, that's what he's proceeding to say, that's what he's going to explain. And then he gets this number. So at that time, the units, international units, were not the same as now, so there is a shift, but it doesn't change the result, obviously. He ends up now with a Reynolds number that is what it is. I'm going to go to figure 3 so that you get an idea of where he goes. And stage is yours. He's comparing 3 organisms. So try to guess what he's going to write. Reynolds number at the top, it's who? What do you say? Yeah? A human being. So a human being inside the swimming pool. Apparently below a swimming pool, because his head is inside. 10 to the 4 means that when he swims, or when she swims, yes, there is inertia that is playing a role. 10 to the 2 for fish, it's also inertia plays a role, so the fish can swim actually efficiently. And you recognize the 10 to the minus 4 I was talking about the other day, which are the microorganisms. So you see that the Reynolds number is panning 10 to the 8th order of magnitude, only because of size, right? Everything is in water. The density is the same, viscosity is the same. Only thing that is changing is going to be the size. So returning to your question of yesterday, it just, that's a size effect, and you cannot escape it. It's like this, right? So he proceeds to say that there is no way you can have an intuition about it unless you go through the calculation slowly. So that's what he does. Now I'm going to go back to the text. So it's an estimate. Somehow we did it, but you see that in this paper, I think 90% of what I said was written. Yes?
Linear relationship between the size
and Reynolds number, yes. That's fascinating, right? Because it's not intuitive.
So fishes, among fishes also maybe some big fish may have
Higher, absolutely. They will benefit from inertia much more than smaller ones. Well, here we go. In figure 1, you see an object which is moving through a fluid with velocity v. It has dimension a. In Stokes' law, so Stokes' law, it's F equals 6 pi eta AR v, which is basically what was on the denominator of the Einstein relation. The object is a sphere, but here it's anything. Eta and rho are the viscosity and density of the fluid. The ratio of the inertial forces to the viscous forces, as Osborne-Reynolds pointed out slightly then than 100 years ago, is given by AV rho over eta, which essentially what I was writing the very first day. It's easier to remember its dimension. For water, it's 10 to the minus 2 centimeters squared per second. The ratio is called the Reynolds number, and when that number is small, the viscous forces dominate. So we are back to business, essentially, of this active matter swimming. Maybe I'll go a little bit through the next paragraph. I don't want to spend too much time on it for the sake of time, but I will read it. Now there is an easy way, which I did not realize at first, to see who should be interested in small Reynolds number. If you take the viscosity and square it and divide it by the density, you get a force. No other dimensions come in at all. So this is typical of scaling arguments or dimensional analysis. You take parameter of your problem, you are exhausted in the parameter of the problem, you don't forget anyone, and then you make the combination of the parameter to end up with the right unit of your system. There is a theorem which is used in math and physics, which is the pi theorem. If you never learned about it, please go ahead and learn it, because that's a way to get a complete description of your physical problem. And this is a dimensional analysis. It's extremely powerful. I mean, usually you cannot escape these values after that. So that's what he proceeds to say. So basically he says, well, you get a force if you make this operation. No other dimension come in at all. Eta squared over rho is a force. For water, since eta equal 10 to the minus 2 and rho equal 1, eta squared over rho is 10 to the minus 4 dim. This was the international unit at that time. That is a force that will tow anything, large or small, with the Reynolds number of order of magnitude 1. In other words, if you want to tow a submarine with Reynolds number 1, tow it with 10 to the minus 4 dim, so it gives the force. So it is clear, in this case, that you're interested in small Reynolds number if you're interested in small forces in absolute sense. 10 to the minus 12 Newton, 1 piconewton is a small force. This is a parenthesis, but just returning to your point of it, it's still physics. I think I wanted to read it. He says, well, what's funny is that there is another part of the world of physicists who is interested into small forces. Is this one. The only other people who are interested in low Reynolds number, although they don't usually invoke it, are the geophysicists. Well, you are totally in another part of, nature. The Earth mantle is 2 pi to have a viscosity of 10 to the 21 poise. If you now work out eta squared over rho, the force is 10 to the 41 dim. That is more than 10 to the 9 times the gravitational force that half of the Earth exerts on the other half. So the conclusion is, of course, that in the flow of the mantle of the Earth, the Reynolds number is very small indeed. Physics. Basically, it means that whenever you have a system well posed, the Reynolds number is generic. It can treat any problem whatsoever, as long as you take, you should stand by this to know which term is negligible or not. So I think several were asking the question, okay, but where is the mass, right? Where is the acceleration, right? Are we talking about, something that makes sense? Yes, yes, and yes. It's just negligible, so then it's out. So far, so good? Questions or no? Okay. I'm moving forward. He makes a move. I think you already guessed what he was doing next, so this is going to be quite easy and quick. Now consider things that move through a liquid. The Reynolds number for a man swimming in water might be 10 to the 4th. If we put it in reasonable dimensions, for a goldfish or a tiny guppy, it might get down to 10 to the 2. For the animals that we're going to be talking about, as we'll see in a moment, it's about 10 to the minus 4 or 10 to the minus 5. For this animal, inertia is totally irrelevant. We know that F equals me, but they could scarcely care less. They don't care. It's irrelevant, which is funny, right? Yeah, he has a lot of sense of humor. If you read the text, it's like Feynman. If you read Feynman or hear Feynman online, you laugh all the time and learn a lot of physics, actually, in a way. I'll show you a picture of the real animals, but in a bit. But we are going to be talking about objects which are the order of a micron in size. That's a micron scale, not a structure, in the animal in figure 4. In water, where the kinematic viscosity is 10 to the minus 2 centimeter per second, these things move around with a typical speed of 30 micrometer per second. Somebody had 90 micrometer per second the other day. Who was it? It was you, right? Maybe you were right, right? You were on the right range. If I have to push that animal to move it, and suddenly I stop pushing, how far will it coast before it slows down? So basically, I'm pushing and then I stop. How far will it go? You make the estimate. The answer is about 0.1 angstrom. This is peanuts. So basically, even though there is inertia, literally speaking, there's going to be a little force that is going, if I push and then no force being applied, but basically it's peanuts, and it takes it about 0.6 microseconds to slow down. I think this makes it clear what low Reynolds number means. Inertia plays no role whatsoever. If you are at very low Reynolds number, what you are doing at the moment is entirely determined by the forces that are exerted on you at that moment and by nothing in the past. Let's dwell on this sentence. It's extremely profound. It's essentially words, but it's extremely profound. It means that in low Reynolds number, history does not count. Okay? What do you say?
Is it a Markovian process?
It's a?
Markovian process.
I need the word.
Markov.
Markov, yeah, Markov, sorry. Markovian process. Basically, it's a process for which there is no memory. So none. Basically, time does not matter. So it's not Markovian, essentially. So basically, time is out. So you need to see that in the next steps, it's absolutely essential to consider it. So step number 1, we are at low Reynolds number. Step number 2, time does not matter at life at low Reynolds number. It's fine? For those of you who are used to the Navier-Stokes equation, which is the essence to write the dynamics of this object, basically when you neglect the term that have to be neglected because you are at low Reynolds number, you have an equation that is independent of time. If you change t by minus t, the equation is the same. So basically, time is out. There's no, time does not play a role. Okay? So now comes the next step. Somebody knows the next step of this paper or not? No? It is called the scallop theorem. Anyone has heard about it? No? So it's a way to remember it. When you hear it once, it's impossible to forget. So this is the, probably this I'm just giving you for the sake of being complete. You see now he has the bacteria that we were talking about, 1 micrometer. These are the flagella in which you have microtubule, 9+2, everything that I talked about. You have the velocity, you have centipoise, you have the low Reynolds number. Everything that he described is right. So then he proceeds to say something that I was proposing the first day. It helps to imagine under what condition a man would be swimming at, say, the same Reynolds number as in a sperm. Well, you put him in a swimming pool that is full of molasses, and then you forbid him to move any part of his body faster than 1 centimeter per minute. Now imagine yourself in that condition. You're under the swimming pool in molasses, and now you can only move like the hands of a clock. If under those ground rules you are able to move a few meters in a couple of weeks, you may qualify as a low Reynolds number swimmer. That's a way to illustrate somehow a metaphor for what it would be looking like for a human being. I was using another example, which was a swimming pool cricket, where you would have the ball at the very bottom, and then there would be no inertia. It would be a very different game, actually. The next bit, and this is something that is going to illustrate what I was repeating over and over again since Monday, or since Tuesday to be more precise, is broken symmetry, basically. And this is going to be essential. It's now. I want to talk about swimming at low Reynolds number in a very general way. What does it mean to swim? Well, it means simply that you are in some liquid and are allowed to deform your body in some manner. You know, when I swim, or when you swim, I don't know which type of swimming you do, but basically if you do this, which I don't know the word in English, you move like this and you do this. Right? So basically that is essentially swimming. According to you, what do the microorganisms do? What do you say? They do this? Anyone else wants to try? Like this? Fair enough. Which is different from what you did. You did this. What is different between you and you? Yes, you are another one. No, it's good if you manage to do them. It's really the point. How do they swim?
Sir, it's essentially should be sweeping, beating, and propelling motion.
Sure, I agree with you. It should be propelling because at the end of the day they are moving. So yeah.
Sweeping a large amount of water backwards.
Yes.
So in any way we can do it. So maybe they do it by rotating their ciliary.
Okay, so if I'm doing this.
Yeah.
So they are doing a chiral motion to make an asymmetric motion.
I could be. I don't know. I mean, I know, but I pretend I don't.
Pushing against water.
Yes.
So the body moves.
Sure. Okay. But basically, if I'm doing this, maybe I do this. If I do this, I can swim.
Yeah, but in the upward direction. If you do it.
So if I'm doing this, am I swimming if I am a microorganism? Who says no? Who says yes? No. No? Yes? Okay? So there is a real debate. Basically, if I'm doing this, can I swim at low Reynolds number? Why? Maybe yes. I mean, essentially, I'm moving water. Imagine I'm doing this very, very fast and this very slowly. Maybe I'll be able to swim. Yes. Okay, so now if I'm doing fast and slowly, I'll be able to swim.
So this is synonymous with how birds fly.
I wish I would agree with you, but you remember the Reynolds number is different.
Yeah.
So that's the key difference. Birds are at high Reynolds number. Microorganisms that we keep talking about who swim are at low Reynolds number. So the physics is different, inertia is out. So if I'm doing this, or let's say this, am I swimming as a microorganism? Yes. Let's read. It's okay, you see the problem, right? It's basically how eventually they're going to swim. So I want to talk about swimming at low Reynolds number in a very general way. What does it mean to swim? Well, it means simply that you are in some liquid and are allowed to deform your body in some manner. You deform, basically, deform the body is basically doing this. I mean, or if, doing something like this. That's all you can do. Move it around and move it back. Of course, you choose some kind of cyclic deformation because you want to keep swimming. So cyclic means I was up, I'm going down, I'm going up, I'm going down. And it does not do anything good to use a motion that goes to zero asymptotically. You have to keep moving. So in general, we are interested into cyclic deformation of a body on which there are no external torques or forces except those exerted by the surrounding fluid. In figure 5, there is an object which has a shape shown by the solid line. It changes its shape to the dashed contour, and when it changes it back, when it finally gets back to the original shape, the dotted contour, it has moved over and rotated a little, it has been swimming. So I'm going to show you what is on figure 5 so that he sees what he has in mind. This is this object, right? So it's doing this type of thing. Moving like this and moving back. Let's imagine there is such an organism that would do this type of thing. And then he finishes reasoning. When it executed the cycle, the displacement resulted. If it repeats the cycle, it will of course affect the same displacement, and in 2 dimensions, we see it progressing around the circle. In 3 dimensions, its more general trajectory is a helix consisting of little kinks, each of which is the result of one cycle of shape change. So far, so good. He's been progressing very slowly, and now he's going to give the so-called scallop theorem. So here we go. There is a funny thing about motion at low Reynolds number, which is the following. One special kind of swimming is what I call reciprocal motion. So reciprocal motion is this. Because reciprocal, I'm essentially coming back, and the reciprocal, I do the opposite of what I did a second ago. That is to say, I change my body into a certain shape, and then I go back to the original shape by going through the sequence in the reverse. So basically, I do this, and I do this, which is what I was proposing before. I mean, I was playing a bit with the speed to make it more spicy, but basically, it's essentially this. I was doing this, and then this. At low Reynolds number, everything reverses just fine. And this is the point which you need to highlight in red, because this is the essence of the phenomena. Time, in fact, makes no difference. There is no history in life at low Reynolds number. Everything is forgotten at every time point. You see where he wants to go? Time does not matter. So time, in fact, makes no difference. Only in configuration. If I change quickly or slowly, the pattern of motion is exactly the same. So basically, I'm a swimming organism. If I do this and this, or if I do this, doesn't matter. I won't move, which is not intuitive. So again, intuition is bad because of what we are used to when we swim in a swimming pool. If I'm doing this very fast, I know that I will go faster. At low Reynolds number, it doesn't change anything. So basically, velocity time is out. The only thing that counts is essentially configuration. You had a question? It's okay?
No.
Okay. So then he continues. If I change quickly or slowly, the pattern of motion is exactly the same. If you take the Navier-Stokes equation and throw away the inertial term, all you have left is the Laplacian term, where P is the pressure. So if the animal tries to swim by reciprocal motion, it can't go anywhere. Fast or slow, it exactly retraces its trajectory and it's back where it started. A good example of that is a scallop. So basically, everyone in the international community will know it as a scallop theorem. You know the scallop. The scallop is this animal. I don't know what it's called in Hindi, obviously, but you recognize it, right? It's this one. What is it called in Hindi? Wait. Scallop? Sheep? Okay, so you know which. What did you say? Yes? Okay, so in French it's coquille Saint-Jacques. I don't know what it is. Basically, I'm just, that's what it's called. I don't know but basically you see what it is. And it is really remembered as the scallop theorem. Why? Because he's going to show that the scallop theorem somehow is basically the only thing it can do is do this. It's squirting water, so it can do only reciprocal motion. Because it does reciprocal motion, it cannot swim. Because it's essentially going in this way or the other way, the speed does not matter. So it is called the scallop theorem of Purcell. I'm going to read it, and then he's going to give a solution. So, a scallop opens its shell, slowly or closes its shell fast, squirting out water. It is right here. The moral of this is that the scallop at low Reynolds number is no good. It can't swim because it has only one hinge, and if you are only 1 degree of freedom in configuration space, you are bound to make a reciprocal motion. There is nothing else you can do. The simplest animal that can swim that way is an animal with 2 hinges. So I'm summarizing where we stand. Because organisms are in low Reynolds number, for swimming, I can recapitulate the experiment for crawling. A cell in our body would crawl, but the same would apply. Same problem type of question is that reciprocal motion will not allow a cell to move, whatever regime it is, swimming or crawling. It is the scallop theorem, to remember it. If I do this slowly, fast, doesn't change, I won't move. So I need to have 2 degrees of freedom. Okay? Last bit. He proceeds to say then, okay, fine, so how can I do that? Well, he's going to invent an animal. The simplest animal that can swim that way is an animal with 2 hinges. I don't know whether such animal exists, but figure 7 shows a hypothetical one. This animal is like a boat with a rudder at both front and back and nothing else. This animal can swim all it has to do is to go through the sequence of configuration shown, returning to original as L05. So I'm going to show you the animal that he invents. This is the animal. So it has, two hinges like this. So I'm not sure I'll be able to do it the right way, but basically here, I'm here now, and I have two hinges. And also, instead of doing this, I'm doing this, and then I'm going to do this, then I'm going to do this, then I'm going to do this, and then I'm going to do this. You want to try? It's okay. You see the point? What is the difference between this and this? Yes.
So if you were to do that,
Yes.
it moves sideways.
Because? The question that you ask is a very sophisticated one, that somehow you have already predicted in which direction it will move, and I think this is still an open question since 1976. He gives the answer of where it moves, but the direction of motion of this actually is a tricky problem. Could someone explain to me why this one will be moving? What is different between this and this?
Seems like it will torque,
Yes.
but not the force.
Because?
It's just like if I give the analogy, there is a rod,
Yes.
and you put in the electric field,
Yes.
parallel to the field,
Yes.
or maybe at some angle,
Yes.
the force filled by that rod to the opposite polarity is not zero,
Yes.
but it will fill the torque. It try to
I 100% follow you. I agree with you. But what is triggering this event, basically? But formally speaking, I also have two hinges, right? So I'm just saying that here I don't move because basically time does not matter, configuration matters. The fact that now I'm having this and then I'm doing through this cycle, it's a real cycle because I return to the original point. This point is the same point as this one, right?
In case of one inch, the force
Yes.
exerted by the object and on itself
Yes.
is always symmetric. But in this case, that is not the case. The direction changes, and that allows the motion to.
Exactly. So basically, configuration is going to change, and that I hope this is ringing a bell of what I was keeping saying since Tuesday. Is that by doing this, the broken symmetry is at play. Okay? This is really the key element. Is that in order to have a motion at low Reynolds number, you need 2 degrees of freedom, and you need to tweak these 2 degrees of freedom in such a way that the symmetry broken is going to happen. This is another way to formulate the chiral statement or the arrow that I was showing before. Basically, any broken symmetry of the filamental or motor filament interaction, ensemble of filament, ensemble of motor, and everything that I talked about so far is at the core of motion. Okay, please.
if you look at S2, for example,
Yes.
and imagine that to be in water.
Yes.
So the pressure difference is along the axis that
Yeah.
is connected to
Yes.
therefore, it's only possible that you move along that direction.
I see what you mean. Now the point is that I invite you to follow. I see the argument, but the thing is that I invite you to follow some lecture of Jean-François Joigny in France, professor au Collège de France, where in fact he treated this problem in one of his lectures. And you're going to see that your intuition is probably right, but the counter-argument is also right. Somehow, because I think he proposes Purcell in his paper in the direction in which it goes, but it's not very intuitive at all, actually. The thing that is absolutely true is that you need to break the symmetry, you need to have a chirality, and as soon as you have a chirality, it will eventually move. So I think you got the point. Somehow, this will not move. If I'm doing this in cycle, I will start moving. So the game is to chase chirality in all this type of motion. Another way to see that is to have θ1, θ2 axis, which is the angle of the hinges. And you see that S1 has a θ2 that is negative, a θ1 that is positive, S2 is here. So you go through a cycle. And if you go through a cycle, then you are back to business. And basically, you can have 2 degrees of freedom and you will start moving because of the broken symmetry. It's fine? Good. So we should stop at 11:15? Yes. So I have time for one more point, and then I'll show Howard Berg presenting his experiments, right? So where am I standing? I'm saying that this paper from Purcell at Life at the Reynolds number is basically postulating most of the key points that are still relevant until today. I mean, there were new features, obviously, but he postulated them in a very clear manner. All the microorganisms, the cells, are in a life at low Reynolds number. They have to respect this rule and to overcome this rule. And the way they can move eventually is to break the symmetry through a chiral structure, through their motion, by construction. And the way to remember it for the rest of your life is scallop theorem. Usually it's working. Remember scallop theorem, tuk, it's not working. Right? It's unforgettable. So basically, this is what you have to track each time you see a motion inside the living matter. The last bit that he presents is answer to your question, partially. So that's why I wanted to show it. I invite you to read the paper. It's accessible online. So you see, I mean, he's starting to have all the organisms that we saw under the microscope. E. coli, Spiriculum volutans, Chromatium hockini. There is also another one that he shows a little bit later on, I think, which is here. This one, I think it rings a bell also, this one. You know, you had this one under the microscope that was looking like a corkscrew. Yeah, that was, I think all of you had it somehow. I don't know its name, to be quite honest, but it was moving fast. Pretty efficient, right? It was like jingling around. And you see, this is a broken symmetry there, right? There is a helical pitch, and so the conformation is different, and by this is allowing somehow the statement that time does not matter, configuration does. So the last bit I wanted to share was this one, because it turns to a question that you were asking, which is coming back to chemotaxis. So later on, he talks about an experiment by Howard Berg, which is, I'm going to, in 5 minutes, I'll show you Howard Berg talking. He passed away recently, actually. Try to guess what this is.
Like a trajectory, sir.
Yes, trajectory of Tuesday afternoon, right?
Brownian.
Yeah, so it's not Brownian.
Oh, is it directed?
Yeah, it looks like there are some spots where it is directed and some point where it is tumbling. So this is a typical E. coli motion, which you saw in the water on Tuesday afternoon. You didn't have a large range, so you saw it very locally, but you could see sometimes the bacteria was moving very straight, and other phases, there was some tumbling. And basically, he proceeds to say, okay, now I know that it has to have a broken symmetry to allow it to motion, but then what he wants to ask is, where is the good place to stay for food? Which is what we were discussing, because, okay, right, as we saw yesterday, well, a bacteria will have all interest to dwell in a place where there is a lot of food, because that's where it's good to be. So in that strategy, what the good strategy for the bacteria is to do what? To stay in the same place, is to make a lot of tumble, right? Because if you make a lot of tumble, I'm doing it one more time, basically, if I make a lot of tumble, I'm remaining around the table, right? Whereas if I'm moving straight, I'm moving away from the place. Okay? So he had one statement, and this is the last scaling law I want to share. He's asking the question, and this was a discussion we were having yesterday. How fast and how far should you go to discover a new environment if you are in a bacteria? The question is simple. I'm a bacteria. I'm having my environment. I'm measuring the food which is around me because I have receptor. And I want to know whether or not, as we say in French, if the grass is greener somewhere else, right? If there is some better food somewhere else. How far should I go?
Maybe 10 times the body length of the...
It could be 10 times, it can be 100 times. You see, you know, we can get, you know, guesstimate, but maybe it's 10. You know, you have a guesstimate that could be right, but you see the problem is that having an idea, which I think you were talking it as a speed yesterday, or when you go to Christina Marchetti's paper, we were having this private discussion. So he's asking a similar question, but in more a biological point of view, is basically, what is the scale. It could be 10 times, it could be more, but that's what he's going to estimate. So I will finish by this, and then again I invite you to read the paper. It's at the end of the paper. He's comparing steering versus diffusion. Time for transport by steering, type transformed by diffusion. Steering works, so these are the expression. LV over D is superior for 1 is steering only if it is under this regime. And then he makes an estimate and says this ratio is much smaller than 1. Local steering accomplishes nothing. Steering, you know, is like you are steering the local material. So basically, when locally the bacteria is steering, it doesn't do anything. And then comes the last paragraph I'm sharing now. There is some neutrons. There is a diffusion constant. And now the thing is that if you want to increase the local food supply, what is the velocity and the distance that you should go to? And his estimate is right here. To outswim diffusion, L has to be superior to D over V. If D equals 10 to the minus 5 centimeter squared per second, V equals 0.03 centimeter per second, L has to be superior to 30 micrometer. You were not far. Right? You were not totally far. The size of the bacteria is about 1 micrometer. So if you want to discover something new, you have to go above 30 micrometer, otherwise it will be the same environment, because all the objects are diffusing out. If you don't swim that far, you haven't gone anywhere. Why? Because your local environment has the very same composition. I like this argument because it's a way of saying that because everything homogenizes very quickly locally, the bacteria has to go that far to eventually discover a new environment and then make a comparison between the concentration that it was once upon a time in this spot and what it will have somewhere else. The beauty of this scaling argument is that it's a way of saying that somehow, if you know the speed and if you compare it to the diffusion of the medium, it's a way to tell you when I'm not tumbling, if I'm moving straight, I'm a bacteria, I need to go that far to discover something new. So returning to the discussion we were having yesterday afternoon, if you have a bacteria that goes very, very fast, you will rescale the argument accordingly for the length. So it's an interesting exercise that somehow it's a way to compare different bacteria of different speed and to see what is the length at which they travel directionally when they travel to eventually compare concentrations. You see the point? I'm going a bit fast here, but the only point I'm trying to make is that how far should the bacteria go to discover a new environment that is potentially good? Since diffusion is all around, it has to outcompete the diffusion. Does it answer part of the discussion we were having? You see, the point is that if you need to go far to discover something new, otherwise the diffusion will be too much. So all this part is summarized and is explained in this part of the text, actually. It's whenever he's quoting the figure 19 here. At what time I thought that the reason the thing swims is that if it swims, it can get more stuff, but because the medium is full of molecules, the bug would like to have. All my instincts as a physicist say you should move if you want to scoop that stuff up. You can really solve the problem of diffusion in the velocity field represented by the Stokes law around the sphere for its accretion method. And then he proceeds to make the calculation that I showed to you before. The increase in text varies like the square root of the bug's velocity, so the swimming does no good at all in that respect. But what it can do is to find places where the food is better or more abundant. That is, it does not move like a cow. This is grazing a pasture. It moves to find greener pastures, which is essentially what I was telling you before. In the last 10 minutes, what I would like to show is Howard Berg, that he's quoting actually essentially his experiment in the end of this paper per se, who is describing the experiment itself. And then we'll make a pause and we'll start on other topics right after. So far, so good? Okay. So the key message I was trying to convey is that a typical old paper, quote unquote, 1976, contains 90% of what I said this week. It's there. It's still correct until today, and that is worth any nature science cell, right? It's basically this will be true in 1,000 years. It's it's rock solid science. Any questions? It's okay. Good. So I'm finishing by Howard Berg. So this is Howard Berg.
It is as a series of little thin sticks, and if the stick moves sideways, the viscous drag on it is about twice what it is if it moves lengthwise. And if you work that out, you discover that this filament, when rotating, will generate both a thrust and a torque. So as the cell swims from left to right, as shown there, the cell body rotates back. To balance the torque required to rotate. And the thrust generated by the filament is balanced by the viscous drag on the head as it moves through the medium. So everything's fine. Now, if you don't believe in rotation, here is a wonderful experiment done by Mike Silverman and Mel Simon back in 1974. You just break the filaments off of a cell and you bolt the cell down by one filament, and then what happens is the cell rotates. So the motor, instead of driving the filament, drives the cell body, or as it were, the boat. To illustrate that, I brought a mouse here, which has red pants, and it has rotary arms. So if you wind him up and hold him by the tail, the arms go around. But if you hold him by the arm, then the mouse goes around. So this is a tethered mouse. It's a smooth mutant because it only rotates in one direction.
That was the clockwise and counterclockwise of yesterday, right?
Tethered cell technique. And the other thing you can see in this movie is that the little guys are spinning really fast, and the big guys are going around slowly. So in this domain, the motor generates constant torque. So if you increase the load on it by making the thing you want to rotate large, it slows down.
Okay, then.
Now, there's a very peculiar thing about.
So then what he's going to do is that he's going to describe essentially the paper of Purcell. So he's going to go through that. And the last bit I want to show in the last minutes, I mean, you saw what he did. Basically, this is the experiment I was showing to you yesterday about clockwise and counterclockwise adaptation to aspartate and so on. That was this type of experiment. And then to show you that time does not matter at low Reynolds number, this I wanted to show you because it's also unforgettable once you see the experiment once. So he's coming with a syrup, essentially, which is this experiment. And you will see, actually, if I didn't lose the track, actually. So I'm going to play it again. It's right here.
Motion of highly viscous. This, the fact that reciprocal motion won't work. It's a beaker with a cylinder in it that you can rotate, and there's a gap between the cylinder and the beaker that you can put in a bit of dyed. We use glycerol because the Reynolds number, so-called, is small at low Reynolds number, life at low Reynolds number, and the Reynolds number is a length L, a velocity V, density rho, and a viscosity eta. And so if you have a macroscopic model, the distances are large, the velocity is large, and the density of the fluid is just the same. But so you need to use a fluid that's quite viscous to get that ratio to be small. So in this experiment, we use glycerin, which has a viscosity about 1,400 times that of water. So what we're going to do is put a stripe of colored glycerin in the gap between the inner cylinder and the outer beaker. Whoop. Disaster. One of the risks of the experiment is you end up being blue. Okay, you can see the stripe, and it wasn't meant to be at the top there, and that won't work very well, but what we're going to do is mix this up. So you can see the guy coming around. It's funny, it's kind of slanted. Now, I can stop and think and talk and do whatever I want. I don't have to do this continuously. I just have to do it sufficiently slowly that the Reynolds number remains small. So now it's reasonably well mixed. Now we'll unmix it. We go back the other way.
Reciprocal motion. Okay.
Again, it doesn't have to be continuous. It just has to be reasonably slow. Now you can see it's beginning to clear. There we are, look at that. So this is life at low Reynolds number, and this is a very dramatic demonstration that reciprocal motion doesn't work. E. coli is figured out by using helical filaments to beat this problem and live a rich and long life. So in summary, I tried to tell you a little bit about the history of this subject, which is colorful, and about some of the physics that E. coli has had to learn in order to survive in the face of very severe constraints, to be able to swim at very low Reynolds number, to be able to count molecules in its environment and decide when all is said and done whether life is getting better or worse.
Right. So you see the point is that somehow you don't need a sophisticated experiment to convince yourself that reciprocal motion goes to nowhere. I like this experiment because it's easy to do. You could do it in your kitchen, right? You can show it to your friends or nephews or children, right? It's fun because then everyone, you expect that somehow you're going to, impossible to return to the original. But basically this is super convincing that somehow reciprocal motion goes nowhere. Questions? Yes. Yes.
Are there cases where diffusion alone is more beneficial or significant than swimming for microorganisms?
Where diffusion is beneficial. Diffusion not of the neutrons, but diffusion instead of directed motion. So basically, would there be a case for which microorganism diffusion would be beneficial in contrary to directed motion? That's a very good question. I would need to think more, but my immediate answer is probably no. Because in diffusion somehow, you can essentially, you travel random. It allows you, if you wish, to explore configuration of space and time that you would not necessarily do otherwise. So you're going accidentally to go to some spot that you wouldn't have visited. But to my knowledge, but it's probably wrong, but to my knowledge, my immediate answer is no. That somehow the add competition connected to the 10 kT of Tuesday and the directed motion is generic and universal. In biology, one has to be careful when one says this because there is always one counterexample somewhere that is showing that what I said is wrong. That would be my gut feeling somehow, as far as I know, but I'm happy to be proven wrong.
So there is another question. Is inertia relevant? If inertia is irrelevant, what describes microorganisms like E. coli or few cells manage to move efficiently?
If diffusion is irrelevant, that's the question. If
is inertia relevant?
No, inertia is never ever relevant. That's the key message in Monday.
Then he's asking if inertia is irrelevant,
yes,
what describes microorganisms like E. coli or few cells manage to move efficiently?
So these are the molecular motors of Tuesday. I'm not sure the person who is asked followed the former lectures, but in one word, what I try to describe is that there are some molecular motors which are essential to allow the motion at the single motor filament level, and collectively that's the lecture of Wednesday. regulated by the lecture of Thursday, allows them to move straight, and that's what Howard Berg is showing. At the time where Berg was presenting his paper, this molecular motor knowledge was already there, but not as developed as today somehow. So the molecular motor is the short answer. Energy out of equilibrium physics, I mean, everything that I described in Monroe.
So there's another question. Do systems at low Reynolds number stores memory of past motion?
Well, it's a very good one. That goes back to the discussion we were having. If bacteria needs to go to a place which has more food, it has to store memory, but it's not the motion which is the storage of memory. It's essentially could be the transmembranous protein I was showing to you before, the key A, B, W, Y, Z, I mean all the key protein of the signaling network I was showing. The way the receptor-ligand interaction is going to be a kind of local memory about the environment. So basically, depending on the ligand-receptor interaction and the clustering of the receptor, there would be a memory of the local state of the cell, which is going to convert into a motion. But basically, it's an interplay between the sensory mechanism around the bacteria and the motion that's going to be the memory effect. Physics alone, as I said before, time is irrelevant here. It's a mixture of sensing together with adaptation to the sensing.
So then he's asking that, so time reversal stands true for all cases at low Reynolds number.
I'm sorry, I didn't understand the question.
Time reversal stands true for all cases at low Reynolds number.
Time reversal, I mean, time reversal is not at play. I mean, the key message is that if you reverse time to minus t, it doesn't change anything. That's what Purcell was writing, only configuration matters. So basically, time reversal, that's basically this experiment with the syrup. He was reversing time. He came back to a perfectly transparent fluid. So time reversal is irrelevant. You don't get anywhere with this. You need to break symmetry. In essence, at the scallop theorem and everything that I described. I think we need to go, Manoj. So we recapitulate that.
We'll be back in 20 minutes.
Okay, 20-ish minutes. So
basically,
yeah.
yes, go and meet the Vice Chancellor. So we'll come back in 20 minutes and then again we'll continue.
Okay.
Also, we need to do a couple of things. We'll distribute certificates today. Also, we'll have a group photo. So all you will be here. So we should probably extend this session beyond 12 o'clock.
Okay, so see you soon.
So we'll be back. It's just a key now. So you can go around.
Of what I was presenting before with the Purcell life at low Reynolds number. Because we wrap up, and because at the end I think I would like to end with science, I just first of all say it was a pleasure this week, right? A real pleasure for me, that welcoming from Manoj and Soban. It was really outstanding, and your presence, each of you. Your dynamics and enthusiasm was communicative, and so it has been a real pleasure to be with you this week. You're welcome to contact me. I could be slow in answering, but I'm trying to do my best to answer if you send me email. So I'm giving you my email. So my name is Daniel Revelin. I mean, you know that, but I just want to. And so my email is Revelin. It is my last name at unistra.fr. So please, don't hesitate. Again, I'm not sure I'll be answering on the spot, but I'll do my best to answer as much as possible. If you could write a title in your email, which is very clear, you write JNU, so that basically I know that it's coming from you. And it could be anything. It can be questions of any type in systems biology or active matter, or it could be preprints. I mean, many of you are already scientists, established scientists, so it could be any papers that you can think about that you would like to share, or preprints. I'll be happy to read them and to get back to you as much as possible. So again, don't hesitate to send email, and I'll try to be responsive as much as possible. Another point that I want to share, and this question was asked also to me, we are not very good in, I'm not very good in websites, and this you could see that. I'm a more old-fashioned person with paper, and I apologize, but that's how it is. So if you want to know some of our publication, I just show one website where it's approximately up to date. It's not really up to date, but this is this one. And because the question showed up in the T actually at some moment, so it's my last name.igbmc.science. There are some papers which are not reported there. Most of them are on bioRxiv, and it recalls more to the research part actually. This is going to appear on the screen now. And so the rotation I was presenting on Wednesday, as well as the global motion of many cells, epithelial cells in the scientific talk, as well as the directed motion of single cell, most of them are here on this web page that is going to appear at some point. And the way it's working is that you simply go through this page, and if you click, you will have access immediately to the paper. So it's, again, approximately up to date, not exactly. So if you're interested into our work, just you click on it, and that will give you the... If you have a hard time accessing to the journal, I have to say that the journals at the moment are very often blocked. So we put everything on blocked, meaning that you need to have the access, institutional access. So systematically, we put all our paper on bioRxiv. So basically, you will have the very same content on bioRxiv, so you don't need to go to the... I mean, I know there are other ways to obtain the PDF, you know it also, but I will just advertise bioRxiv, right? So that's for this. And the last element, because also the question was asked during the week, and then I'll go back to the lecture. So most of what I was presenting this week, in fact, was matured between 2010 and 2015 when I was coming back from sabbatical. From New York, I was arriving back in France in Strasbourg, and we launched a cell physics master, which is a one-year training for students who finish their master one. And I'm just showing you that so that you know the website. You have excellent training here, so keep your training here. The idea is really not that you go out from this great place, but basically just so that you know it is existing. It's a year program where, in fact, we train students for 1 year in physics, chemistry, biology, math, in the spirit that what I was teaching this week somehow. So this is the website. It's lphysics-master.com. And when you go through it, you can see the program. So you see scientists who are involved in the way we've been tuning the program. So it has been matured in the idea that, how can we run a program for people starting from scratch? The idea is that we recruit after Master 1, because the idea is that the students should get a very strong scientific identity in math, in biology, in physics or chemistry before going to interdisciplinary field. And if you travel through the different items, you will see that in the program, you have detailed, you have basics at the beginning of the year, which essentially give the background to everyone so that people can follow in physics, chemistry, math, biology. This is September. And from October to February, there are some heavy physics, heavy biology, system biology, chemistry, and math integrated in the same scientific direction. So that's the whole idea of the program. So you have the details which are here on this page. You have also actually the faculty members. So you will recognize some of the names because I think some of you know already pretty much some of this community actually, but you're going to see them appearing on the screen. I want to mention John orally because you might know them. There is Carsten Kruse, who is in Geneva. He's a prominent theorist on the field of active matter. Lennart Hilbert, liquid-liquid phase separation. He came from Dresden and he's teaching. He's in KIT at the moment. Other faces that you can recognize, Morgan Madec was the numerical simulation. That's what he was sharing so that I could share it with you yesterday afternoon. Nacho Molina, that was a set of coupled differential equation. Guillaume Salbreux, also from Geneva. Raphaël Voiturier, etc., and so on and so forth. So basically, all the faculty are coming from different places in Europe for this year program to eventually, share this passion for interdisciplinary thing, biological physics. So this week lecture was a kind of best of, I don't know how to call it, but basically extraction from what collectively we've been building as a common knowledge. And then after that, students go anywhere, basically. It's 1 year program, and then they project anywhere in the world to perform their PhD. If you want to know more about this, there is also here, and this is the last bit I want to share, it's in the basics here. We recorded a week program, which is everything is recorded here. Maybe I can make the publicity here for Thomas Lecuy. It's an online course that you can follow, which is totally in English. Here is the physics of morphogenesis. I'm selecting it because this is what I'm going to talk about now. And at that time, if it's working, what you're going to have is the sound and the presentation of Thomas, where he was talking essentially.
Good afternoon, everybody. So it's a pleasure to continue this session on morphogenesis. So my course is not going to be Mr. Wood, which were.
So basically, you see the style. It's an hour of lecture, which allows you to have a feeling about whether or not it's along your taste. And you can travel to the origin of life, microfabrication and biology, collective effect. I mean, many of the topic I was alluding to during the week. So that's the website. Basically, just to know that it is existing in the landscape. Okay? Good. I'm closing the parentheses, and I'm going back to teaching. Okay?
So you apply directly to the.
Yes, you have a contact here, and here on the contact you enter the contact item. The only point is that it has to be done very, very much in advance because there is all administrative, steps for visa that has to be done in advance. So basically, if at some moment some of you or your friend consider it, think about it ahead of time because there is all types of procedure that has been to be done online with Campus France. It's with deadlines.
So annually, once in a year, this is open, right?
That's right. So to make it very, very caricatural, if you wish, for most of the countries aside from Europe, it's better to apply before December or even before November. So basically, the sooner the better because the paperwork means visa. So basically, the visa can be obtained only before a deadline that is very early in the year. It's like December something. So if any of your friends or you are interested, it's better that you apply, before September, say, so that there is a first contact, an interview, and then, if you are eventually selected, then there is the deadline of Campus France. There is no other way to obtain the visa at the moment. It's really like the real deadline. Good. So this was, again, a parenthesis, and I would like to go back to science. So the last, it's okay? Any questions, comments? Fine? Good. So, as I said, the last session I wanted today to talk about classics, in the sense of classics. So the first hour was about life at low Reynolds number. Now I'm going to talk not directly about an old paper, but in the way I'm going to talk about an old paper. And I thought, it's the last moment that we exchange, so I thought it would be the opportunity to talk about it. So it's more like you're going to see that I'm going to really talk about something old. And contrary to what I did so far, which was short talk, now it's going to be essentially on PPT, right? So. I was showing the lecture of Thomas Lecuy, and basically what I would like to do in this last hour, it's a topic that I haven't talked about because of time limitation. It was the physics of morphogenesis. So basically, how does a shape comes about? It's a vast question, difficult to treat in such a limited time, but at least it's an introduction. So I'm going away from the single cell and I'm going to come to the idea of a tissue. And in a way, it will allow to revisit the idea of what is active matter, what is living matter. And I'm going to ask you many questions and I'm going to try to challenge what you said. So the first, I mean, what do you hear in physics of morphogenesis? What do you expect to hear?
Sir, genesis means the production.
Yes?
Morpho means the overall shape.
Yes. So basically, it's going to be the production of shape and how is it going to happen. I do agree with you. Physics?
10 kb.
I like it. Yeah, when you hear physics, because the result could be the biology of morphogenesis, right? So basically, it's an option to treat it as the physics of morphogenesis. So basically, it's a way of saying that you expect to have a force balance, consideration that would imply the generic rule of symmetry, conservation rule, force balance that are at the core of the reasoning, right? So the first point I want to defend is that it's an idea that is old. It didn't start, 10 years ago or 20 years ago. And to go through that, I would like to ask you the question, what is the difference between inert matter and living matter? I'm not talking active matter, I'm talking about living matter. How do you know that something is alive? Just think about out of the box. You never, you want to know if something that you have in front of you is living or not. What would you say? What do you say?
Movement.
Who said moving? Oh, said moving, yeah, moving. Yeah, okay. I think that's seems to be like moving. It seems to be the core of something that we define. So in fact, this idea was expressed already by Aristotle. Just a way of saying that this idea is not from yesterday. He has this say, which is the Greek philosopher, famous philosopher. He writes, "From what has been said, then it is plain that nature, in the primary and strict sense, is the essence of things, which have in themselves as such a source of movement, for the matter is called the nature, because it is qualified to receive this, and processes of becoming and growing are called nature, because there are movements proceeding from this. And nature, in this sense, is the source of the movement of natural objects, being present in them somehow, either potentially or in complete reality." This is philosophy. So, with philosophy, you need to take the time in front of the sentences and to try to say, but in one word, what is he saying? Movement. So movement, a source of movement they have in themselves. What is the difference between me pushing the chair and nature? Internally driven. That somehow it's the nature spontaneously, there's going to be a motion that is going to. There is another part of the sentence which is important, processes of becoming and growing. What does it mean, becoming and growing? Yeah, proliferation, potentially, and becoming. What do you say? Birth, yeah, if you wish. There was nothing and then there is something, right? There is another meaning of becoming, right? Reproduction, I agree with you, it could be growing also, reproduction, or becoming could be also something. Change shape, yes? Differentiating, right? So it's becoming in a sense that you are a stem cell, and after the stem cell state, you change and you are to another state. Eventually, you transdifferentiate to a state that you were before. So basically, this can happen. I mean, probably I'm overstretching what he was writing, right? But somehow you can feel in the words that he's using, being a philosopher, that he's using very, very specifically the words to define actually what nature is. There is a last point I want to highlight, and this is the end of the... And nature in this sense is the source of the movement of natural objects being present in them somehow, either potentially or in complete reality. So this comes often in Aristotle. But I would like you to try to rephrase it with your scientific background. And nature, in this sense, is the source of the movement of natural objects being present in them somehow, either potentially or in complete reality.
Anything that can be considered living to any extent will have an intrinsic force that will move it or that will grow.
Agree with you. So it's internally driven. It's going to be spontaneous. I 100% agree with you. What I would like you to try to dig into is either potentially or incomplete reality. Potentially or incomplete reality. What does it mean?
Potentially means it is an expression of something inside. For complete reality means it is there.
Yes.
Yes. So it is an integral part of existence.
Okay, I like it. So complete reality, it is here. It's happening. So basically, complete reality, this is moving now. So basically, it's now, it's happening. Potentially means it might.
It has the potential.
It has the potential. So what does it mean it has the potential? Could you phrase it with something I was talking about this week? What do you say? It has its own energy, which is not yet used for, but that potentially will be used one day for.
So it has some free will.
No, this I will say a firm no, because the connotation of free will somehow is a connotation where, in fact, if I decide to take this piece of chalk, I have decided to take this piece of chalk because I see it, and this is an expression of my free will. You could even contest that this is free will when I do this, right? But in the case of a microorganism, here I think it's much more neutral. It's potentially meaning it is existing inside the system, but it's not yet used for. I can convert the category for stem cell or differentiation. It can potentially change its state. Everything is present so that the transcription factor machinery is going to get expressed so that the cells change state. But it's not yet in complete reality because it is potential, right? It is not so. In the system biology realm, it would mean that everything is there so that the cell, that the stem cell, become a cell of a heart, but it's not yet there.
Depends on the environment.
On the environment or the genetic program or the path in which it has been evolving, it depends on all these factors. So again, I'm overstretching what Aristotle was writing just for the sake of illustrating it, but somehow most of the words that he's writing are somehow quite relevant to the definition of nature. Yes?
Can we say that a seed
Yes.
is under potentially and
Yes.
completely empty will be soiled?
It's if you wish, it's an observable relation with what you say and what the stem cell and differentiation I said before. It's along the same process associated with growth and transcription factor turning on. Exactly, we are in the same path. So you see, that's how Aristotle mentioned it. Now, I wanted to mention that, and now I will be a bit quicker, and that will allow me to introduce another part of movement. I want to challenge it at this part and returning to a question that 2 or 3 of you were asking before, saying what is different between inert matter and living matter. Well, you remember the motility assay experiment where the filaments were moving straight on myosin. Is it living? No, it's a biopolymer, right? Out of equilibrium, moving in a directional manner. So movement, per se, it's not a living object, right? So what is the threshold between a living object and a non-living object? And that's I would like you can propose some elements somehow, but the frontier is extremely difficult to define because somehow, again, directed motion, it's synthetic chemistry, you can do it, or or molecular motors know how to do it. It's not by the same token that you have already a living matter. Anyone wants to challenge? Yeah, please, yeah.
If I say it's a combination of multiplying and directed, what would that be?
I would argue that somehow you could imagine a pure passive system, and I will show that to you in a moment, with foams, where eventually you would have proliferating forms that would be purely passive because it's foam, and eventually not leading to any type, a foam is passive, right? It's not an active matter whatsoever. So I could imagine a system purely foamy that would be passive and not living.
I'm saying that a combination of directed movement and repetition.
Sure, imagine if I'm trying to build a synthetic tissue or a synthetic cell, I would put molecular motors inside the foam, I would allow it to divide, I would allow it to move, it would be a passive system on which I've been adding directed motion and energy out of equilibrium. By the same token, will I call that a living object? There is an international competition at the moment to create a synthetic cell, and inevitably, most of these people actually are going through this type of reasoning. Is that what do you need to add to a membrane of course molecular motor, of course 10 kT, of course energy, and what do you need to add up to have a living object? What I'm trying to defend is that this definition has its limit because the frontier is a very subtle one. It's a difficult one. Okay? So this being said, I'm moving forward. The cells move. I've been telling that since the beginning of the week, so now I think you are convinced. I'm going to show you some movie. The reason I wanted to show that to you is that, I talked about it during the talk, the scientific talk. What we saw under the microscope was swimming. It's easier to see swimming because, first of all, it's found in the water pond, so and second, it's quick, so you can see with your bare eyes. Cell crawling is a different type of physics, but I wanted to show it. It's an illustration of the fact that the cells are moving. But they are moving in the body all the time. And they are not swimming, they are crawling, meaning that they're going to attach on the surface and they're going to move in a given direction. So everything that I said before about life at low Reynolds number is still correct, but the physics is radically different. So cells move. Everyone is familiar with this type of image or schematics? You raise your hand, the ones who know? Okay, so I should describe it. Yes, okay, good. So I should show it very quickly. This is a mammalian cell, a fibroblast spread on the surface. It has a triangular shape. You have here so-called stress fibers, so actomyosin, again, many filaments, many motor, which are anchored on the surface. So basically, the cell is going to do this. It's a force dipole for the most physicists. So I have a force dipole here, another force dipole, another force dipole. And this is the shape of a triangular cell, and at the front of which there is a so-called lamellipodia. This is actin filament I was describing on Wednesday morning. So this is a lamellipodia, and at the front you have the so-called filopodia, which are actin filaments which are parallel. If you have a hard time having a feeling about what this is, actually, I think I took it, and I hope I took it, but I think I did take it. If you think about the cell, actually, we are having fun in the lab, as you see. If you take a cell, which is so this is a cell. So it's difficult to blow. This is a cell like this, right? Imagine I have a cell which is a sphere. Basically, a filopodia. Imagine I can put my finger inside, which I cannot because if I do it, it will inflate. But basically, if I put my finger inside, I will be able to deform the membrane because I put my finger inside, right? So this finger is about the structure. You see that the physics of this object is not totally obvious because if I take this cell, if I put it on the surface and I force it to be spread and to make this deformation, I mean, you played with this when you were kids, actually. I play, even though I'm not young anymore. But basically, if I do this, you see that there is an immediate problem, right? Because it's not going to remain like this, all right? So the cytoskeleton is capable of going from this shape into a flat shape and eventually a triangular shape, right? So on top of that, there is exchange of the force between what the cytoskeleton does and the surface to eventually allow the cell to move at low Reynolds number, so force being applied at every time, all the physics I was mentioning before. Back on the envelope, this part is allowing the cell to probe. So now there is no story about rotation and tumbling frequency, it's a total different story. Here it's the cell which is capable of basically sending protrusion and all kind of event everywhere. When there is a point that is good for the cell, whatever that means, it's going to anchor somewhere and propel in this direction if the cell is polarized. Okay, so that's the cell, individual cell. The regulation that I was talking about yesterday comes into the game. And these are the GTPases rho. Anyone knows the rho GTPases or not? No. Yes. Good. So what is the rho GTPase that regulates stress fiber? You know. Rho, basically. Rho regulates actomyosin stress fiber, RAC regulates the lamellipodia, and CDC42 regulates the filopodia. So you see that already for the cell motion that Aristotle was talking about, and I'm making a big stretch again here, is that if you take a single cell, it has the force component of the stress fiber, lamellipodia, and philopoda under the regulation of small GTPases, which are going to turn them on and off. So the raw GTPases will turn on and off the stress fiber, turn on and off the lamellipodia, turn on and off the CDC42. It is much more subtle, but what I'm trying to say is that it's a combination between these two approaches which can describe fairly well a cell. It's active physics together with systems biology. That's a bit what I'm going to convey as a message. So I'm boiling down the sand to the minimal. You have here stress fiber. You have the points of adhesion. So it's my balloon, which is the cell anchored on the surface and then moving like this. And eventually, with this, I'm ready to make a motion. So I'm not going into the detail of the regulation, but if you wish, it's a typical pathway which looks like the repressilator type of pathway I was showing, but now for the fourth generation, where Rho activates Rho kinase, inhibits myosin light chain phosphatase, Rho kinase activates myosin light chain, there is contraction and the force being applied. So we are in a network of turning on and off the force. What does it look like? I showed this movie already earlier this week, so now you are familiar with it. The movie actually is here. You see the cell moving. So it's really, think about the cell as a balloon, right? And try to make the balloon move. It's fun, but it's difficult. But you see eventually that the cell gets polarized. Eventually, after polarity, it's moving in the same direction of polarity and then switching gears. It was a question online, I think, that was asked the very first day, or maybe the second day. You see here that the cell gets polarized now. It's moving in the same direction as its polarity, switching its polarity, and then the polarity velocity are aligned. Switching polarity become isotropic, and then again having another polarity and moving in the direction of the polarity. The time is extremely slow. We're talking about 33 hours. So what is the speed of the cell? Back on the envelope. You know, we are not very rigorous here. We are, reasoning like back on the envelope calculation. So in 30 hours, how much distance the cell have been doing? 200. Let's make our life easy. Let's say 300. So 30 hours, 300 micrometer, we are at a speed of 10 micrometer per hour. It's fast, it's slow, it's what? Very slow. Compared to the 90 micrometer per second, that some of you measured in the pond water, we are really out. 10 micrometer per hour seems to be very, very slow. Let's think about this number. What is the size of a cell?
10 to 15 micrometers.
10?
To 15 micrometers.
Yeah. So it means that in 1 hour, the cell will have moved its length. It sounds to be something, right? It means that the cell has been moving its own distance with this typical time scale. Now imagine a cell that is embedded inside the blood vessel, because the cell is larger than the blood vessel in which it is, and it has happening in the body, in a cancer system, where eventually a cell would have escaped, let's say, for the colon and would travel in the blood vessel to eventually being in a vessel that is extremely small, so the cell is blocked, actually. In 1 hour, it has been performed in 10 micrometer. If it is polarized in 10 hours, it would have done? Yes? 100 micrometer. In 100 micrometer, you enter into a tissue. In 100 micrometer, you enter into the lung. So basically, in 10 hours, it's in the lung. In 20 hours, meaning in a day, it's really deeply inside the lung. It has performed already a distance that is sufficiently large. And then it can dock, and after that, start to proliferate. So if you put in perspective this time scale of 10 micrometer per hour, you realize that it's not particularly slow. In the lifetime of a person, somehow it means that within days, a cell that is invading another tissue is about this time scale and this, of invasion and eventually dwelling inside the tissue. So putting it in perspective, this speed is not very slow. It's for the human time, it's becoming relatively fast, actually. For the anecdote, I have some colleagues in France who've been organizing, on a lighter note, an international competition of the velocity of cells. It's like, I don't know what you call it, it's Formula One. It's the car which are going the fastest. So they organized the competition, it was lines, and people were sending their cells all over the world. And the idea was tap, and then how fast, what would be the cell that would be the quickest. You cannot go much quicker. I mean, you can go maybe 100 micrometer per hour, but much further. Again, bear in mind that you are bound by the physical laws associated to it. So somehow you cannot go much further away, whatever cell you could consider. Yeah, please.
Force the cell to move in the desired direction to be in a race.
Yes, this is a very good point, actually. The cells that were being sent were placed on a line. This was Manuel Théry and Mathieu Piel. They were put on fibronectin line, and they were putting the cell here at the beginning, making some elements so that the cells were polarized. And then they were moving straight on the line because this line were lines that were adhesive. It was fibronectin. Like the red motif for my scientific talk. So it was fibronectin, the cells were anchored on it. And then by triggering the polarity, the cell could move on the line, and then that was T0, and this was T end. They were measuring the time, the length, and they had the speed. But the point that I'm trying to convey is that somehow this is a typical order of magnitude that you would find that is pretty reasonable. I'm insisting on the fact that I'm talking here in this last session about crawling cells. I'm not talking about swimming cells. Here, and the main limiting factor is the friction with the surface. Whenever you move, maybe so that those of you who are not familiar with friction, everyone bikes in the room? Yes? Okay. Is there friction when you bike with the surface? Yes? It's good or it's bad for your motion?
Good.
Oh, bad, right. Okay, so when is it bad? Yes?
It takes a lot of energy.
Yes, basically if you have lots of friction, if you don't have an electrical bike, you could feel it because then you need many lot of effort, right? Another place where it's very cumbersome to have no friction, which can be dramatic, right? No friction, it is when it is raining. When it's raining, what's happening? You slip, right? So then you don't go straight. So there is a compromise between having sufficiently large friction so that you anchor to the surface and not too large friction so that you can still move. So friction is something of that type. Imagine friction at low Reynolds number. This is what I'm talking about. So I'm talking about the crawling cell that when they move, they need to anchor to the surface and need to anchor to be moving, but overcome the friction with the surface to eventually move. So it's a compromise of that type that I'm talking about. Okay, good. So this is what I wanted to say. And so basically in this context, I would like you to imagine that the cell is going to adhere to the surface, get its polarity, crawl and move, and we using stress fiber to apply a force on the surface, lamellipodia to search for the environment, anchor somewhere else, move, anchor somewhere else, move under the regulation of rho GTPases, and eventually move, and obtain the velocity that I mentioned before. I'm going to pass this because I presented it already in the scientific talk the other day, but you can rectify the motion in a way that can be reproducible. I'm just showing you the movie quickly, and then basically here you have this is partially answering to your point. If you take triangle shape, you can direct the cell motion that eventually is going to move straight. Here you see the adhesive domain that were on the surface, and eventually the cell move in the direction that you want. Yes?
So, here I understand that there is some surface.
Yes.
But inside our
Body?
Mm-hmm. What do you mean by surface? Is it a set of tissue or?
So, yeah. Between the organs in the body, what you have is extracellular matrix protein. So it's a 3D network of entangled protein which have a reticulant. I think it is called in the soft matter community chemical gel. So you have the polymer and you have the cross-links between them. So the cells, which are fibroblasts at their name, are going to crawl in 3 dimensions, anchoring into this gel and pulling like this all over. So this is, if you wish, on 2D flat surface, but this is formally the equivalent of a cell moving into a 3D network. That's formally the same. And the velocity, we and others are making experiments in 3D with the gel, you end up with about the same. The physics is a bit different, but conceptually the concepts are pretty shared. So I'm summarizing where I stand, and I'm trying to explain why I was using this back to Aristotle. It's just to say that cells move, not more, right? Basically, I can recognize living matter by saying it moves with the limit I was saying before, that what is the frontier. Nowadays, you can prepare synthetic molecular motors. I was mentioning Ben Feringa, Stoddart, and Jean-Pierre Sauvage, Nobel Prize for the synthetic molecular motor. We were talking about it with one of your colleagues. So basically, molecular motors are around, the frontier is difficult, but nevertheless, the fact that cells move is a given. Active matter, living matter is moving, and that's already was foreseen by Aristotle. Now I'm going to come to an old classics paper, and that was the argument of this morning, to tell that many of the ideas were already formulated ways back. So this book has some outstanding points, some limitation, but at least it's the outstanding point I would like to share for now. It was written by Darcy Thompson, so it was a book that was quite old, and the book was published in 1917 and republished in 1942. I'll show you one example of the absolutely extraordinary way the book was written, and this is here. Darcy Thompson wants to compare the tissues with foams. He says, well, you know what foams are, right? It's water and soap. When you wash your hands, essentially you generate a foam. And he's comparing 2 states. The state here with 4 bubbles or with 4 cells, and the state here with 4 bubbles and 4 cells, but a difference between the left and the right. And I'm asking you the question, what is the difference between the left and the right? Shape. More surface area in one case than the other one. Fair enough, okay, good. All what you said is good. What else? What comes to you when you observe the left and when you observe the right? Yes? This one is symmetric, the other one is not. Yeah, this one is symmetric, this one you see that there is a change, or there is something which has changed, yes? And what you were saying? Yes, this one, they are not in contact. Very good, right. According to you, which one is stable, which one is unstable? Do they have the same stability or do they have a different stability? Come on. You were saying something? Which one is stable, which one is unstable? If you stay over time, it remains the same or it disappears. The first one is? This one is stable, this one is unstable. No. Reverse. Who says this one is stable? Okay, fair enough. Who says this one is stable? Okay, who has no opinion? The majority. Good. It's written. That's another way to go for it. Yeah. Okay. But you see that there is something funny, is that if you think about it, it seems a very easy problem to think about, both in terms of forms and in terms of tissues, because the tissues are made of cells. And a priori, Darcy Thompson had, the sufficiently careful organization and way of thinking about it to compare forms and tissue at this scale. But you have a feeling that it is different. So for those of you who went or who might go at some moment to the physics, I'll show you a movie of forms at some moment. This happens to be a stable configuration. 120 degrees is the configuration. And he was a friend and colleague, and friendship in this interdisciplinary field is very important because then you discuss about, relaxed way different topics, which allows to go into this realm. So he was very friend with a very good physicist in this field, and so they started to think about this problem. And so here you see the 4-cell configuration happening. So let me show you what he writes about stability and instability, and you will see why I'm focusing about it. He writes here this paragraph, which each time I'm reading it, I like it because I think it's very well formulated. As a matter of fact, the simple cross is no very rare phenomenon, even in the frog's eggs. So he was observing the eggs under the microscope and then drawing the shape. But it is a transitory one and unstable. Viscosity and friction, you see viscosity and friction again? They are back to business. Viscosity and friction may enable it to endure for a while, but the partitions inevitably shift into the stable three-way configuration. So the cross is unstable, and inevitably it's going to go to this organization of Furler. And then he says, and that's something that's also striking, in such a case, the pole of Furler manifests itself slowly and as it were laboriously, but in the more fluid soap bubble, it does so in the twinkling of an eye. You know, twinkling of an eye is when the eye is doing this very quickly. So what he proceeds to say is that if you see that in a foam, you won't see it somehow. It will immediately disappear. So I don't know if you knew that before, but basically when you observe a soap bubble, when it is like this, it will disappear immediately. Whereas 120 degrees, it will stay for a while. So he's contrasting inert matter of foams and active matter of tissue and saying there is a striking difference, which is the stability of the cross. So far, so good? 100 years ago, okay? So it's not like last week in Nature of Science, right? It's like an old paper. Now I'm telling you now, and I'm showing 100 years later about a comparison between a tissue and a foam. So this is a foam here, and this is a tissue taken from Thomas Lecuis's lab in Drosophila, but there are many around in the literature about tissues, right? These are cells which are adherent. What do you see on your left about the cell organization? What do you see striking that I was highlighting in the book chapter of Darcy Thompson? Hexagonal organization, meaning that the angle between edges is 120, right? What do you see on the right-hand side? You have, sometimes you have 7, I totally agree with you. Sometimes you have something like 120, right? Sometimes you have more complicated structures, such as here, rosette structure. But basically, there seems to be a common denominator with 120 as a basis, plus something else that is happening. Of course, it's very different. Here you have a gas, and here you have a liquid. Here you have the cytoplasm. Here you have adherent junction with cadherin, beta-catenin, alpha-catenin, the cortex. So it's a radically different system. Nevertheless, the pattern formation has to share some similar features. It's okay? So you see the jump that I'm making now is that saying that Darcy and Thompson postulated the problem with 4 cells or 4 bubbles, and basically in modern area, in most of the developmental biology community, they consider this statement as being important. They write the equation associated to it, and they try to explain the morphogenesis, the physics of morphogenesis, which is the title of what I'm trying to talk about now. So I need to add one last aspect, and then I'm done. When you study the physics of foams, you stumble into 2 transitions. So I know we've been having a week of physics of biological systems, so it's a lot, but it's the last 15 minutes, so last effort. You have 2 transformations, this one, which is called the T1 process, and this one, which is the T2 process. Can somebody explain to me why T1 process is called neighbor exchange? Yes?
I think in the first one there is a share of center.
That's right. Absolutely. Very good. So you got it. So basically, if I'm drawing the cell, in the first case, I have cell number 1, cell number 2, cell number 3, cell number 4. It's going down like this. I'm going to have 1, 2, 3, 4. If I'm going down, I'm going to have here 1, 2, 3, 4. So basically here, on the first case, I had 2 and 4 that were neighbors. And if I'm going down, I have 1 and 3 that are neighbors. So it's a neighbor exchange. You could make the game with the, you are 4 like this, you switch your chairs. That's essentially what's happening. It's a very important transformation, right? It means that somehow you won't exchange the information with the same person or with the same cell. So here, 4 exchange information with 2, and 1 and 3 are far away, whereas here, 1 and 3 are close. You're familiar with the game Tetris? I think this has the same name internationally, so it's a kind of Tetris game where in fact each of the cells is going to go somewhere else and exchange with another cell. The other event that is happening is the T2 process, and T2 process is disappearance. Why disappearance? Yes, what do you say? The middle cell is gone. What does it look for? I mean, what does it look like for the most biologists in the room when the cell disappears? Dead. It can be dead. It's delamination. It's happening in the body, in your intestine all the time, in the crypt and villi, where the cell eventually go out. It can be alive, but somehow it's a cell that delaminates. It goes out from the tissue. So this is T1, this is T2. And when you have T1 and T2 in mind, you can have most of the physics of morphogenesis already intuitively. Wrong, because you need to write equation and everything that I said starts again. But at least the basic building blocks are more or less okay. So I'll show you a movie of foams. It can look a bit funny to show foams now, but this is foams. And I like this movie. It's taken from a colleague in France, actually. If you see my mouse, I don't see it. Yeah, it's starting now. So this is the movie. Look, 4. Look at the 4 here. Look. Tac. 4. Tac. If you spot another one, 4. Look, this region. Puff. Tac. So you see, when you have a 2D form, Darcy Thompson in 1917 was correct, and somehow the T1 and T2 are operating, and when you have 120 degrees, it is remarkably stable. It keeps the angle forever. So T1 and T2 processes are essential for the transformation of the shape of form. Of course, to go deeper, you need to enter into the difference in pressure, the surface tension, Young-Laplace law. I mean, you need to spend some time to learn the physics associated to it. But essentially, the T1 and T2 processes are really at play, and now you see the movie continues to run, and this is remarkably stable and it stays for a while. So I'm showing you this just to highlight the fact that, this physical rule for the forms are solid. 120 degrees seems to be the baseline, and in principle it should be solid for most of the tissues, any pattern whatsoever, even though the physics is radically different. Okay? So T1 transition, T2 transition, the physics of forms, and then adaptation to the physics of tissue so that the physics of morphology is right, is driving many of the international effort in developmental biology. So far, so good? So you see the jump that I was making between Monday and Tuesday from passive particle to active particle. I'm doing now the same, but for passive forms and active forms and tissue. It is the same intellectual exercise somehow. It's that you take for granted ideas that are true for passive matter and you try to change them to understand living material. Okay? So I'm making a big gap voluntarily. And I'm quoting now Eric Wieschaus, and I invite you to look online some outstanding talks of Eric Wieschaus. I was showing before the talk by Howard Berg on iBiology website. That was where it was taken. Eric Wieschaus gives also some talks on iBiology. It's really a nice website where you have really outstanding speakers all over the world who are giving their talks. I know Eric Wieschaus gave a talk there. I just want to mention a talk he gave in 2013. You see the title, On Growth and Form of the Embryo. It's a title that Darcy Thompson had selected but adapted for the embryo, from gene expression to tissue mechanics. Eric Wieschaus got the Nobel Prize about the morphogene gradients, and he decided at some point to say, okay, what I did was important, but now I need to go to another field of science, the physics of morphogenesis. That's not, it's a bit summarized, but that it is in the direction that he's been thinking in the last 10 or 20 years. I just want to read, and actually that will make an echo to what I said the very first day. During development, cells in an embryo face two major tasks. First, they must express distinct combination of genes appropriate for specific cell fate, such as muscle and skin. Once such patterns of gene expression are established, cells must change their shape and position to match those cell fates. These transformations are remarkable for their speed and precision, but also for their incredible beauty. He's very enthusiastic when he speaks, and that's very communicative. Somehow it's very nice. If you want to relax, watch developmental movie online. It's really nice. The associated cell shape changes depend on local patterns of gene activity, but how such patterns are converted into the physical properties controlling shape and motility is a major unanswered question in biology. So even though he's the Nobel Prize winner of morphogen gradient, he says, well, that was good because then we could have a pattern of gene expression establishment, but we don't know how they change their shape and position to match these cell fates. What is he saying essentially? Physics of, that's right, physics of morphogenesis is the way to go. That somehow you can change the pattern of expression, you can change the cell fate, but if a cell fate is not matching the right position, somehow you won't have a functional organ. So somehow what you need to think about now is how such patterns are converted into physical properties controlling shape and motility is a question that now has to be addressed. So that's why I wanted to share that, because, I mean, if you put them back to back, it gives a vertigo, because somehow the motion is intrinsic to nature, but how the motion is going to be converted to make a functional organ is a major unanswered question. So in the last 5 or 10 minutes, I'm going to show you observation, and it's a game, so to finish on something funny. I'm going to show you observation. I'm summarizing, you know, its motion is essential, also at the cell level, multicellular level, and the physics of forms transverted to physics of morphogenesis is the way to go to integrate the biology of morphogenesis and development. So I'm just showing you observation, and I'm going to ask you which organism is it?
One day after fertilization, the embryos...
I'm cutting the sound because that will give you the answer. So what is it? What? A cell. Right. Sure. Eukaryotic. There is a nucleus. I'm sorry? You need to... There are 2 cells now. More. So I'm going to ask you which organism is it. If you wait, it's going to give what? Human embryo? No, I'm asking the question. I'm not saying it is. What? Zebrafish? Mm-hmm. I think you said something at the beginning, but I didn't hear. This is morphogenesis, right? I mean, no doubt it's an embryo. So basically, which organism is it? Ah. So I heard zebrafish. I heard human. I heard... Ah, there is hatching. Right. Difficult to know, but I heard 2 or 3 different hypotheses. I'm going to another one, and I'm telling you what it is. And you're welcome to guess what it is. How many cells do I have now? 2, right? Okay. And then it continues to proliferate, and you're going to see something that is going to be striking, I hope, with what I said right before. I have 2 cells, and then I'm going to have 3, and now I have 4. Oh, and what is the shape? Bubble shape, okay? So the 120 degrees is not an illusion. It's somehow, you know, this is happening, right? What is the organism? Yes, I am like you. No, I have the same skepticism that somehow, what can it be? It's difficult to say. It's very difficult. Anyone wants to guess? Okay, so I let the movie go, and then shout to say what do you think it is. So you have the time below, actually. It is coming from the Tony Heinemann lab in Dresden. This is a very... This is the beauty that Eric Bischers was highlighting. It's simply, it is appeasing to observe these movies. Look at what's happening now. You're going to have, this is compacting a bit, and then you're going to have something else, which is now. Two blobs which are forming, one on the right, one on the left. And here we go now. It is undergoing a very smooth and slight rotation. Okay, which organism is it? Human? Who said human? Okay, you agree that it's different from the former one, but yeah, it's yeah. Okay, now what is it starting to do? It looks like what? What is it doing? I mean, if you would describe with word. What? You said worm? Okay, well, but what is this motion here? It looks like what? What? Time to come out? Yeah. Yeah, but how can it move like this? Yes, but for this you need what? Tuesday lecture. I agree, but even simpler. To move, you need energy, and the energy is going to be ATP, and plus the energy in order to apply these forces. So that you need what? Lecture number 2, Tuesday. You need to add. What was the core of the Tuesday lecture? In addition to energy, you need to have direction, which is going to set by broken symmetry given by molecular motor, right? So basically you have expression of muscle protein, which are molecular motors, which eventually take this contraction together with cell proliferating, changing their state, and then the word of Eric Bischers, they change their state to accommodate their cell fate. So basically they're going to write in the right spot. So as Manoj was saying, it's time to go out. Here we go. Yes, snake, worm, yeah. So, but it went out, right, eventually. And you see that the time here, it's 11 hours. You've been seeing from the very first stage where you had a single cell up to the most latest of development. So it is another organism, which is C. elegans. That is its name. You were right, it's a worm. But which is a very nice model system because it has all the organs actually that are necessary, and there are many Nobel Prizes actually around the usage of C. elegans because it mimics a whole body developmental biology. I'm showing this movie just one after the other to convey the message that this is living matter, right? So basically the roles regulate. It's impossible to say which organism it is. I mean, you were wrong. I was wrong also when I was seeing this movie first. Somehow you need to go into the details of the development, but the first stage is this is simple rules of growth and processes of cell-cell interaction, proliferation, the physics of morphogenesis. That's the essence. I want to finish with a very last word, and then I'm done, really. I don't want to make it overly long. I want to ask you the question, what is the state of living matter? I've been talking a lot about physics of morphogenesis now. I would like you to ask, and again, I'll stop with this, what is the state of the matter? How many states exist in physics usually?
4.
Which one they are? Okay, so what is the state of tissue? What is the state? Fifth one is what? Yes, but I'm ending up with 4 so far. Somebody said 5?
Both Einstein.
I'm sorry?
Both Einstein and
Bose-Einstein. Oh, wow. Okay, fine, fair enough. Yeah. Fair enough. It's yeah. I mean, one of you was talking about the quantum physics world. Here we are really in the classical world, and Bose-Einstein condensation will lead us to another interesting stimulating direction. But it's yeah. So I'm asking the question. The final one is that what is the state of this matter? What is the state of this matter? Gel? So it's solid or liquid? Liquid? Solid? How do you know that a cell or an object is a liquid or a solid? Which experiment do you do?
When we apply the force, it will change the shape of the object will change, then it will be liquid. If it will not, then it would be crystal.
So another way, very empiric way, if you are a teenager and you want to know, yeah, a liquid is going to be take shape of the beaker. That's right. So that's an experiment that was done a while ago by a group. Actually, there are other groups who've been performing it. They prepared the ball of cells and they put it on the surface. Right? If it's a liquid, what the ball of cells is going to do? Yeah, it's like wetting. You have a drop of droplets. If it is a solid, it will remain. So let's run the movie. The focus is at the bottom, actually. And what you can see here is that eventually, what does it happen? It is spreading, suggesting that the sphere that is here is behaving as a liquid. But it's a solid, right? So somehow, how can you reconcile the fact that you have a solid behavior and a liquid behavior? Some of you may know or may guess. Viscoelastic behavior. So somehow in the short time scale, it's going to become as a solid. In the long time scale, it will behave as a liquid. So you have a viscoelastic behavior with a typical Maxwell time, which would be around 10 hours. Good. So this state of matter can be solid or liquid, elastic in short time scale, liquid at long time scale, and you can revisit most of the morphogenetic events with this vision, where you will play with the viscoelastic property of tissue and you will see how eventually they will grow. To introduce confusion, and then I'll definitely stop, is it the privilege of living matter, or is it this type of behavior, is it existing also in inert matter? Again, to play on the boundary between living and passive. So if you never saw that, I brought another game. It's coming not from France, actually, it's coming from the United States. But I think I took it with me. I hope I took it with me. It is called Silly Putty. You know it or not? I'm sure you have it in India, but I don't know what is it called here. It's kind of a clay. You saw this guy here? So it's a clay. Clay, but it's not a normal clay. So imagine this is a ball of cell or tissue or, So I'm not very good in this, but I don't know where it's going to go. So it's a liquid or a solid? Yes. Sorry. It's a solid. You agree it's a solid. I mean, with this I can play as much as I want. Thank you so much. Thanks. So it's a solid. If I'm taking it, if I'm pulling on it, it's a solid or a liquid? You have a doubt. So I'm going to repeat the experiment. I'm taking this. It's a solid or a liquid? Solid, okay. It's a solid or a liquid? It seems like the behavior of the material depends, not exactly the force, on the time scale of the force, so the delta F over delta T, right? The variation of force that I'm applying. Which is essentially saying that this property of living matter exists also in inert matter. And I will end with this, with the threshold between one and the other. This deserves a life process of thinking. Thank you. We'll stop here.
Now is the time to conclude. It's been very nice for the last 5 days that Professor Daniel Revelin has been here with us, and he has shown us a completely different world He has exposed us to completely different world where we were actually imagining many things. We are imagining many things for the last 5 days with him, and I hope that many of you came up with some objective to learn something new, and I hope you accomplished all those objectives. And for this, we should thank Professor Rebellin. He has come all the way from France and stayed with us for 5 days. And I'm sure it's not easy with all the time gap and all. Now is for him, it's about 5-6 hours of gap with France and India. So to go through all these things, food, and still he's energetic, and is very interactive, and I'm getting a lot of feedback from the participants that they are really enjoying these lectures, and so we also should thank our dean, Professor Shobhan Sen, for helping us to run this course for all these days, giving us seminar room and support staff. Also, I should not forget to thank all the participants for being here and participating in this lecture series and enjoying it. And also now is the time I think we should distribute certificates. So it's the last thing that we want to do. Also, I would like to thank all the students who help us, all these 4 years to communicate with participants and making arrangements, right? So some of the students help us as volunteers, like Pooja is here, Pooja Choknar, Kritika is here, another Pooja Negi is here, Pramod is here, who was helping us, Pooja Dahiya was here, Shashank was here, and all others. I'm forgetting any name. So all these students were really in the front for helping us in the lab, also helping with the microscopes and everything. So those are the ones who actually are behind this scene, running the show. So let us get the these certificates. Who is having the certificates? You have to bring. So it was got enough time to conclude in a nice way.
Thank you.
Or you needed more time?
I'm good.
It was good.
I mean, I don't know if I'm good, but at least what I wanted to convey.
Yeah. We also want to have photograph in the end with all the participants for everybody's memory.
With pleasure.
Yeah.
I think this topic, you can treat them 10,000 hours.
Yeah.
And it's still
if somebody wants to speak something about the, is welcome, he or she.
You must be tired.
Anybody wants to say something about the course, so you're welcome to say.
Sure. Should I keep the mic or not? It's okay. You can hear me.
Done.
Okay. I think it's no. I think I have it.
You have?
Yeah.
Like how you demonstrated the clay.
Yes.
You said that very
Depends
well.
on force and the time.
Yes.
But if we go at the low Reynolds number, if we talk about the microorganisms,
Yes.
so we are excluding time there. So how do we define the state of?
Okay. So this is a very interesting question. In fact, I'm going to translate what you said at the scale of a tissue. Can you hear me at the back, or I put the mic? You can hear me? Okay, good. So basically, if you are at a scale of a tissue itself, imagine this was the clay, right? Imagine I have a tissue here and another tissue here inside an organism. And there is one tissue that is getting formed because, I don't know, there is a somite in somitogenesis that is getting formed. I'm taking a random example, right? And there is another somite. The somite are the places at the back which are present everywhere from the snake to... basically, you have here one somite getting formed, another somite getting formed, and in between you have a tissue, right? It's not void. There is a continuous matter. This is the extrapolation of what I was showing. Because basically you have two tissues which are pulling apart. They are pulling apart with a given time scale and a given length scale, which gives a speed. And this is associated with a variation of force over time. I am at low Reynolds number, right? There is no acceleration. You see what I mean? But nevertheless, the tissue in between will react very differently depending on the time scale. If I'm going very quick, the cells in between won't have the time to collide with respect to each other. It will be too quick. So the only thing they can do potentially is to break. If it's going very slowly, the cells that are in the tissue in between will have the ability to glide and eventually secure a continuum. You see, the point is that, and then there is no acceleration. It's a pure viscous problem of force and viscosity. Another example, sorry, I'm getting too long now, but there is another example, which is a cancer. In cancer, probably if those of you who are familiar with cancer, there is a process that is called viscous digitation, where you have two tissues, and there is one tissue that is invading with the shapes of fingers. You can see that in many cuts online. If you make, this is, if you wish, the similar type of physics, where you have, energetically, it is more favorable to grow fingers at the interface, and it's at low Reynolds number. Every tissue will locally apply a force that will lead to a given velocity, and the force balance locally will lead energetically favorable the growth of tissues. And you multiply the example for every process. It's exactly the point. It's okay?
Thank you. Punching is safe? Online. Anvesha Datta?
So actually you have to sign.
Oh, sure. Maybe I will go on the... Thank you.
Thank you.
बाद में
Delight.
Thank you so much.
Anuradha
Thank you.
Raj.
Thank you.
पूजा पहले दे दो फिर साइन करा दो
फिर बाद में कराएंगे। नहीं सर, जो है अभी करा लेते हैं, बाद में जो नहीं है उनको एक साथ कराएंगे। हाँ वो थैंक
Thank you very much.
आदित्य चक्रवर्ती, आर्यन त्यागी,
Aryan,
आशीष,
Thank you very much.
छवि तोमर, चिन्मय नंदा, दीपक कश्यप,
Thank you.